Hi and welcome to my OCRA A-Level Biology revision with me, Christine. So today's lesson I'm going to look at the erythrocyte and the role of the hemoglobin inside the erythrocyte and the oxygen dissociation. So I need to take a step back and look at erythrocytes. Now, we know that erythrocytes are differentiated cells. We know that they have a very specific job to play in which they are transporting oxygen around the body. And we know that they are continuously made from hematopoietic stem cells, which are multipotent and found in the bone marrow. Now, the reason they're continuously made is because they only actually have 120 days worth of their lifespan. And that's because as they differentiated to have their biconcave disc shape, to increase that surface area and volume ratio, they actually removed the nucleus and all the organelles inside the cell that were no longer necessary for the role in which it has to transport oxygen. So before the cell was mature enough to leave the bone marrow, what needed to happen was it needed to make sure it had all the necessary compartments and then remove anything that was no longer required. So this means that the cell is small. It also means that it's flexible in shape. Because it doesn't have all the organelles, it therefore means that it is able to carry much more hemoglobin, which is necessary for that transport of oxygen. But it also means that it can squeeze through capillaries. It means that it's now more in contact with the capillary wall. By increasing that surface area that's in contact, it also results in a short diffusion pathway. So it's important that we note the differences, but before we move on, I want to bring in two extra differentiation parts which are necessary for the transport of carbon dioxide. So also inside the erythrocytes are high concentrations of an enzyme called a carbonic anhydrase. Now we know it's an enzyme because it says A's at the end, and the carbonic anhydrase is a necessary enzyme for the transport of carbon dioxide. Well, it's an enzyme which is a protein that came from protein synthesis. So therefore it was produced before the removal of the nucleus, because the nucleus contained the chromosomes which contained the genetic information which led to the production of that enzyme. Another thing that happened was the differentiation resulted in ensuring that there were transport proteins on the cell plasma membrane which are necessary for chloride shift. Now, the reason I've gone into this amount of detail, and do please check out all my other videos on enzymes, on protein structure and differentiation, but you have to look at biology, especially A-level biology, as the bigger picture. Everything is interlinked. So when you start to look at it as a big picture, it should hopefully start to make more sense than just looking at each individual module on its own. So what I want to look at now is hemoglobin then. So hemoglobin is a red pigment. Now it's a conjugated protein. So it's a protein. So I know that it is made up of 4 polypeptides. So I know its protein structure is a quaternary structure because I have 4 different polypeptide subunits being held together. Now it's called a conjugated protein because it also, for every polypeptide subunit, it has a prosthetic group, which is a non-protein group attached. That is what the heme group is. So the heme group is a non-protein part, which we call a prosthetic group, which is attached to each subunit. So that's why it's called hemoglobin. Now it is a globular protein, and therefore we understand that when hemoglobin inside your blood is fully saturated, what we're saying is that it has joined 4 molecules of oxygen, and we call that oxyhemoglobin. So when the oxygen is being added to the hemoglobin, that is called loading, and we say that that is association. if we are going to remove and therefore unload the oxygen from the hemoglobin, then that is known as dissociation. So this is a reversible reaction that can occur. Now, for every one hemoglobin molecule, 4 molecules of oxygen can be bound if the hemoglobin is fully saturated. Each erythrocyte has about 300 million hemoglobin molecules. So therefore, this makes it a very efficient molecule for transporting the oxygen and a very efficient cell for transporting the oxygen around the organism. Now, it's important to note that although it can bind 4 molecules of oxygen to it, it's important that we understand that the first oxygen that binds is actually the hardest oxygen to bind. After the first oxygen has bound, that will actually change the tertiary structure of the protein and changes the affinity, the ability of it to bind the oxygen to it. So as each oxygen molecule binds to the hemoglobin, we end up having an increasing affinity for oxygen. So to understand where partial pressure comes into it and how the dissociation curve looks like, we need to go back to, well, what is the oxygen concentration inside the atmosphere? So partial pressure is the term that we use when we discuss the concentration of a gas in a mixture. So if we understand that the atmospheric air contains 21%, which is oxygen, we therefore would say that the partial pressure of oxygen in the atmosphere is 21 kilopascals. So the partial pressure of oxygen at the lungs that we have just ventilated is about 21 kilopascals. If we were to look at our cells, which are all around the body, our normal cells, they have a lower partial pressure of oxygen. So the partial pressure of oxygen at normal cells is between 5 to 7 kilopascals, but when we look at very active cells, then it's about 3 to 5 kilopascals. So if the concentration of oxygen were decreasing, so would the partial pressure. So we can show that in the dissociation curve. So if we look at the oxygen dissociation curve, we can quite clearly see that our percentage saturation of hemoglobin is on our y-axis and our partial pressure of oxygen is on our x-axis. And the shape is always a sigmoidal curve. And that's because that binding of that first oxygen is always the hardest. So that's why it's a slower slope, a slower curve at the start, because it's hard to bind that first oxygen. Therefore, what you get is the affinity being increased the more the oxygen binds. And that's why you have an increase and then it levels off into a plateau when it gets to that fully saturated place. Now it normally becomes fully saturated at about 14 kilopascals, and that's why you end up with the oxygen in the alveoli being able to diffuse across into the blood, because there is a change in that concentration gradient. The blood that's being pumped to the lungs is deoxygenated, and that deoxygenated blood is therefore going to have a partial pressure which is greater in carbon dioxide and therefore much lower in oxygen. So to understand what happens at the alveolus then, we understand that oxygen is at a greater partial pressure. So oxygen is going to diffuse into the erythrocytes. It's going to diffuse down its concentration gradient, and therefore once it gets inside the erythrocyte, it will then bind to the hemoglobin. Now the hemoglobin is a buffer for the blood and therefore normally has hydrogen ions bound to it already. So when the oxygen comes in and binds to the hemoglobin, that hemoglobinic acid, that will therefore dissociate the hydrogen ion. So the oxygen will bind to the hemoglobin and the hydrogen ion is released. Now, hydrogen carbonate ions will be pumped back into the erythrocytes up at the alveolus. Now, that means that those hydrogen carbonate ions and hydrogen ions can now come back together to form what's known as carbonic acid. That carbonic acid will then dissociate, and therefore the carbon dioxide and water that is produced as products means that the carbon dioxide can now diffuse out of the blood into the alveolus. So it's important to note that the oxygen coming in and the carbon dioxide moving out, that is a form of diffusion. The oxygen diffuses into the erythrocyte and the carbon dioxide diffuses out. But there's a whole load of reactions occurring inside the erythrocyte. And it's important that we understand what they do and how that helps in that transport of oxygen and carbon dioxide. So the oxygen and the carbon dioxide diffusing from the alveolus to the blood capillary or from the blood capillary to the alveolus, that is gas exchange. The transport of the oxygen and the carbon dioxide is how it's bound to the hemoglobin. So if we look at the respiring tissue then, we know at the respiring tissue that we would have our carbon dioxide being released through aerobic respiration. We have carbon dioxide and that will then diffuse into the blood. Now that carbon dioxide is going to bind to the water, and when it combines with water, inside the erythrocyte, there is lots of carbonic anhydrase. So remember we talked about the carbonic anhydrase before. Well, this is where the enzyme is needed to produce that carbonic acid. That carbonic acid is then going to dissociate into hydrogen ions and hydrogen carbonate ions. Now those hydrogen ions and hydrogen carbonate ions, they are dissociating into the ions, which is going to have an effect on the pH as well as the ionic balance within the cell. Now to ensure that the blood plasma acts as a buffer, hydrogen carbonate ions are pumped out of the erythrocyte and chloride ions are pumped into the erythrocyte. Therefore, we are pumping the same charged ions, one out for one in. Therefore, we're maintaining the electrical chemical balance inside the erythrocyte, and that is known as the chloride shift, where one hydrogen carbonate ion is pumped out and one chloride ion is pumped in. Now, the hydrogen ion that was released from the carbonic acid when it dissociated, that is going to now bind to the hemoglobin. Now, obviously, we have pH. If we are decreasing the pH because we now have free hydrogen ions, those hydrogen ions can now bind to hemoglobin, which is a protein. That pH decreasing is going to affect the tertiary structure of the hemoglobin, and that is therefore going to result in the hydrogen ion binding to the hemoglobin and the oxygen being released, and that oxygen can now diffuse across into the tissue fluid and then into the respiring cells. So it's important that we note that the carbonic anhydrase is an enzyme, so it's a biological catalyst which is going to speed up the rate of reaction by lowering the activation energy. But it cannot work without a cofactor. So carbonic anhydrase is an enzyme which needs zinc, which is the cofactor which allows it to be able to function. So it's important that you do note that you have to know zinc as the cofactor for carbonic anhydrase to allow the erythrocytes to be able to transport carbon dioxide. The carbonic anhydrase needs to be able to speed up the rate of reaction of the carbon dioxide and water into carbonic acid. So what we therefore get is that the carbon dioxide and the oxygen have been exchanged, so gases exchange, and then the erythrocyte has now bound the carbon dioxide so it can transport it around to where it needs to go, to the lungs, for it then to be released back into the atmosphere. So it's important to note that carbon dioxide can actually diffuse straight into the blood plasma, and the reaction to carbonic acid and the dissociation can also occur in the blood plasma, but it doesn't occur as efficiently as it does in the erythrocyte. So about 5% is actually carried in the plasma when we look at the carbon dioxide transport. About 20% is carried, specifically bound to hemoglobin, which is known as carbaminohemoglobin, and 75% of carbon dioxide is carried in the form of hydrogen carbonate ions. So it's important that we understand that the hydrogen carbonate ions act as a buffer so that that carbon dioxide can be transported, and it can therefore allow for the cells to still function as they need to. So if we go back then to our dissociation curve then, and we note that this is what's known as the normal dissociation curve. Well, we know that if we have very active cells, we will decrease our concentration of oxygen, and therefore the partial pressure of oxygen will be lower. That decrease in oxygen concentration is because the oxygen has been utilized for respiration and the carbon dioxide has been produced. So when there is a high partial pressure of carbon dioxide, because the respiring tissues have produced more carbon dioxide through the process of respiration, that is going to lower the pH. That lowering of the pH is going to promote that dissociation of the oxygen. The hydrogen ions binding to the hemoglobin, changing their tertiary shape, is therefore going to affect its ability to hold on to the oxygen, and therefore this causes the dissociation curve to shift to the right. So when there is an increase in carbon dioxide at the respiring tissue, when there is a high partial pressure of carbon dioxide, that therefore results in what's known as the Bohr shift. The dissociation curve shifts to the right, therefore meaning that the hemoglobin is going to release the oxygen at a partial pressure. That means that those respiring tissue are able to get that oxygen that they require. So the Bohr shift is a shift to the right because we have a high partial pressure of carbon dioxide at the respiring tissue. We have therefore lowered the pH. So it's important to remember carbon dioxide and water produces carbonic acid. The carbonic acid dissociates, releasing the hydrogen ions. The hydrogen ions are going to lower that pH, therefore promoting that dissociation of the oxygen. Now, if we look at fetal hemoglobin then, what we see is actually a shift in the opposite direction. So in the placenta, the partial pressure of oxygen is much lower. Because the fetus is not able to breathe on its own, it's in the amniotic fluid, it has no access to oxygen if it's not provided by the mother's placenta. And therefore, in the placenta, the fetal hemoglobin, so the fetal blood, and the maternal blood come very close in contact but do not mix. So what needs to happen is that the fetal hemoglobin has to have a higher affinity for oxygen. Therefore, the oxygen is going to be transferred from the maternal adult hemoglobin. That is going to dissociate, and that oxygen will therefore move and diffuse across the placenta into the fetal circulation and bind to the fetal hemoglobin. So that causes the curve, the shift to the left. So if it is Bohr shift, the dissociation curve will shift to the right because we have a higher partial pressure of carbon dioxide. We're changing the pH. When we're looking at the fetal hemoglobin, it's going to shift to the left because the fetal hemoglobin must have a higher affinity for oxygen. The other thing they can also ask you in the exam is they may give you information about myoglobin. So myoglobin is the oxygen molecule binding within your muscles. So it is a protein which is found in your muscles. Your muscles do the respiration. They need to contract. They need to do lots of respiration, and therefore they need to have a higher affinity for oxygen than the adult hemoglobin. So it's important that the key thing, if you are looking at any other organism, or if they're giving you any other example, which they love to do with this type of question, that you are aware that if there is a lower partial pressure of oxygen and the hemoglobin is binding the oxygen to it, i.e. the curve has shifted to the left, you would therefore say in the exam that whatever that hemoglobin may be, that has a higher affinity for oxygen. If it shifts to the right, you would say it has a lower affinity for oxygen. So do make sure you know the difference. And if you like this video, then please do click on the like button and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com. It will help you to revise and give you questions to test your knowledge on this.